Am I audible and visible?
Yes, Navanita, you are audible and visible.
Okay.
Yeah.
So, good evening, everyone. A very warm welcome to all of you to today's international webinar on biotechnological approaches for environmental resilience, insights into sustainable food packaging and stress tolerance in plants. This event has been organized by the IQAC Research Cell along with the Department of Zoology and the Department of Botany, Jangipur College, as we proudly celebrate the 75th anniversary of our institution. Jangipur College, established in 1950 in Murshidabad, West Bengal, has long been a beacon of higher education for the region, providing invaluable opportunities primarily to the backward rural population. The college offers undergraduate programs in arts, science, and commerce and has been affiliated with the University of Kolkata since 1999. We are also proud to hold a NAC accreditation of B++ in Cycle 2. In addition, Jangipur College has been offering postgraduate courses in select subjects under the DODL scheme of the University of Kolkata since 2016-2017. In the spirit of academic excellence and social responsibility, today's webinar aims to address critical global challenges related to environmental resilience. Through innovative research in sustainable food packaging and plant stress tolerance, we look forward to explore viable solutions that can positively impact both our environment and society. Before we begin, I would like to thank our principal, Dr. Prarthita Vishwas, the chief patron of today's event, for her constant guidance and support, as well as Dr. Basudev Chakraborty, IQAC coordinator, for his valuable role in shaping today's session. Special thanks to my colleague and joint convener, Dr. Shombu Mukherjee, and the organizing team for their tireless efforts. Now, I would like to invite our principal, Dr. Prathita Vishwas, to deliver the inaugural address. Over to you, ma'am.
Yeah, good evening to one and all.
Hearing?
Am I audible?
Yes, ma'am, you are audible.
Yes, ma'am. Good evening, one and all. First of all, I like to thank Navanita for the deliberation and the moderatorship which she is doing. At the same time, I welcome you all, each and every participant, in this one-day international webinar, which is a very important aspect and the topic which will be discussed by the two resource persons are very eminent in this time or of today's present era. And at the same time, I would like to state that we are actually entering into the 75th year of Jangipur College, and this is a part of this process. I welcome the first speaker, Dr. Preetam, as well as the second speaker, Dr. Ruchishmita. both will be spending their valuable time, and they will be deliberating on very important issues, which we will be, of course, listening. And I hope that the students will also be listening, and they will be also having a knowledge about the topics which the resource persons will be discussing. So I wish the webinar all the best from my side, and at the same time I wish that everyone comes back with a takeaway from this webinar. And I would also like to congratulate the research cell of Jangipur College, IQAC cell, as well as the departments of botany and zoology, who have given their very best in preparing this international webinar. So together we form a team, and of course this webinar will be a grand success, which I believe. So I hand over this e-podium to Navonita again, so that she can give the details and start the first session. Thank you, one and all.
Thank you so much, ma'am, for your inspiring words. We know that environmental resilience and sustainability are critical. Biotechnology life and proof plant and cellular by enhancing plant resilience and promoting sustainable
ম্যাম, আপনার কথাটা ঠিকঠাক আসছে না, মানে বুঝতে পারছি না আপনার কথাগুলা।
Actually, Navonita, your voice is very clear. We are sorry for some technical issues, so we will start the session very shortly.
Extremely sorry for the interruption. Am I audible and visible now?
Yeah, it's okay now.
Yeah, extremely sorry for the
Yes, ma'am.
interruption. There is a network issues. Uh, So in this context, it is now my great pleasure to introduce our first speaker, Dr. Preetam Sarkar, Associate Professor in the Department of Food Process Engineering at the National Institute of Technology, Rourkela. Dr. Sarkar earned his PhD in Food Sciences from Purdue University, USA, and has made significant contributions to the field of food packaging and biopolymer-based materials. His research focuses on valorization of agricultural byproducts, developing bioactive packaging system, and creating sustainable food-grade emulsions and gels for antimicrobial and probiotic delivery. Dr. Sarkar has an extensive publication record with recent studies in biodegradable nanocomposite films, bio-nanocomposites, and innovations in food packaging. He has presented his work at various prestigious international conferences, and his innovative approaches have earned him several academic awards, including recognition from the Institution of Food Technologists. Today, he will be sharing his insights on sustainable food packaging system from agricultural byproducts, the case of tamarind kernel polysaccharides. This promises to be an enlightening session as we explore the role of natural biopolymers like starch and xyloglucans in developing biodegradable packaging solutions, a crucial step toward mitigating the environmental impact of plastic solution. Dr. Sarkar, we are excited to hear your thoughts and learn from your expertise. Over to you, sir.
Hello, can you hear me? And can you see me?
Yes, you
Hello?
also audible and visible, sir.
Yeah, I'll just share my slides. So thank you to Professor Navanita Mukherjee and thank you Professor Shoma Mukherjee for the wonderful invitation to Jangipur College and the 75th year of your establishment of the college. And it's a great time to speak about sustainability and as we move towards more sustainable practices. So thank you for the invitation. So I'm just trying to open my slides. I think, can you see my slides?
Yes, sir. Your slide is visible.
Okay. So again, thank you, Professor Navanita Mukherjee and Professor Shoma Mukherjee for the invitation. So the topic of our presentation, so I am Dr. Preetam Sarkar, working as an associate professor in the Department of Food Process Engineering at the National Institute of Technology, Rourkela, which is in the state of Odisha, India. And we have been working with sustainable biopolymers for the past few years now, so almost like a decade, 10 years. We have been trying to explore the world of biopolymers from different types of food products, agricultural products, and co-products and byproducts. So our current focus is more focused on the agricultural byproducts because there are a lot of byproducts, agricultural systems which are being thrown away and they don't have much utilization. So we're going to focus on valorization and the utilization of those biopolymers. Presentation today will be sustainable food packaging systems from agricultural byproducts, the case of tamarind kernel polysaccharides. We talk about tamarind kernels because India is the largest producer of tamarind in the world, and we have a lot of tamarind in this country, but there's not much application and not much utilization of the tamarind seed polysaccharide. So we're going to explore that area. So with that, briefly, we'll introduce to our NIT system. The NIT system, there are, if you know, there are 31 NITs, which are centrally funded technical institutions across India, where each state has one NIT actually. So all these NITs, they are classified as an institute of national importance, and they have we have undergraduate programs, master's programs, and the doctoral programs. So these are public technical institutions, and this is directly under the Ministry of Education, Government of India, with the annual budget of across 31 NITs around 5,040 crore or $600 million for all the NITs located across 31 cities in India. So specifically our Odisha NIT, the NIT Rourkela, is one of the broader and the diverse NIT across all the NITs because we have 20 departments in this in our university, in our institute. And out of that, our department is a comparatively newer department, which has started from the past 10 years. At the Department of Food Process Engineering. So we can see some images from our campus. We have a QS world ranking of around 291 to 300 in Asia and 67 in Southern Asia. And we also stand at 676 overall category in sustainable ranking. So now QS ranking also gives rankings for sustainability index. And in the Times world ranking, we are in 601 to 800 world university rankings and 189 in Asia university rankings. So these are the latest rankings of our institute. So with that background, the research team, so we are talking, so the research goal and the theme of our lab has been primarily focused on biopolymers from food and agricultural sources. We try to explore biopolymers, different types of biopolymers and colloidal systems from food and these agricultural byproducts. If we talk about biopolymers and colloids, they will be ranging from the nanoscale to the micro scale to the macro scale. You can see on the y-axis. And then we have two particular targets. And so when we talk about food, of course, the food safety and the security becomes a very important aspect of food. So in the food, there are four major aspects of food process engineering. There is food processing, food process engineering, food chemistry, and food microbiology. Okay, so I have a PhD in food chemistry, but these are the other three aspects or four aspects of food which are really important, and the pillars of our field. So we have a food safety aspect because we consume food and we have to have safe food. And also, the other hand, if you have to process the product and reach it to the consumer, we have to use packaging, right? So packaging becomes a very important and integral part of the food industry. So therefore, of course, the current standard in the food industry, of course, people are using synthetic plastics. So therefore, that leads to that has environmental safety issues, and that leads to that is not. We are not maintaining circular bioeconomy. So our target is to achieve circular bioeconomy. So therefore, overall goal is to focus on the preservation of food systems. Of course, there are different ways of preservation of food systems. We have systems from the most common system that we use for food preservation is thermal processing. We always heat our food. We cook the food right in our home, in our kitchen, because heat is the biggest source of to make the food safe and preserved, because by decreasing or by reducing the microbial count, the microbial load. Okay, and of course there are other techniques like high pressure processing. There is your pulsed electric field processing. Nowadays we have cold plasma processing, radio frequency processing, ultrasound processing. So many different processing technologies are there, which are also classified under emerging technologies, which are used in the food. Also, we have microwave. Nowadays, microwave is very common. We have microwave processings. So these are all used in the preservation of the food. But the problem is all those techniques, they are very expensive. Okay, apart from the heat, apart from the thermal processing, those systems are quite expensive, and they cannot sometimes, they can also sometimes hamper or damage the quality of the food. So therefore, we got to design we got to think about alternative strategies which can be used to preserve our food systems. So one of that is by focusing on antimicrobial compounds. Okay, so we try to preserve food with natural antimicrobial compounds. We will talk about that as we go on. So therefore, so we try to preserve the food by using these natural antimicrobial compounds. And so the goal of our lab is to focus on translational biopolymer science. So we have so many different classes of biopolymers. We talk about starch and xyloglucans. We'll talk about them. And then we apply, so we take the knowledge, the fundamental science, and then we translate to our food packaging systems. And also we want to understand the biopolymer structure and functions, which can also improve the packaging functions, for example. Okay, and these are some of the research funding that we have. We'll talk about that towards the end. Okay, so again, so if we just talk about the food, or think about the food system, the food system is very delicate. Okay, any food, you see, the food is a very delicate system. It can be under threat of a lot of stress factors, starting off from microorganisms, from light. Light is a very bad. So I mean, light, just because light has ultraviolet, so your UV rays can really degrade the quality of the food, like reacting with vitamins. It can degrade the quality of vitamins, for example. Physical damage, temperature abuse. Temperatures also can degrade food quality. Environmental pollutants, water vapor, mechanical damage, and oxygen. Okay, oxygen is also there, like singlet oxygen, triplet oxygen, which can also react with the food molecules and degrade the quality of the food. So with that, if you want to make the food or keep the food system safe, you got to. So there are two major problems. We are talking about microorganisms, and we talk about synthetic polymers, because the food will come in a package system, and which are generally used by synthetic polymers. So we have food safety concerns, and we have environmental pollution concerns coming up from the food industry as such, right? So if you want to target both these global problems, so these are global problems, right? There are food safety issues across anywhere you go in the world. There is food safety problems, and environmental pollution, of course, because of the food packaging systems, is there anywhere in the world. So we were thinking of solutions which can really, of a single solution which can be used to target both these global issues using a single solution. Okay, so if we just go about certain news releases on food packaging, like plastic food packaging was the most common beach trash in 2018, for example. So this is from National Geographic. Okay, so in plastic food packaging now outpaces cigarette butts, for example, as the most abundant beach trash. Okay, so again, now just a recent study, just a month back, probably this came out that India is now the world's leading plastic polluter, which is a study done by at the University of Leeds in UK, and they have shown that that their data shows that India is the leading producer of plastic in the world, and it's not China. Okay, so China was #1 till last year, but now we are the leading polluter in the world as for plastics pollution. Now, what's the problem with the plastics? Okay, so if we talk about synthetic plastics, the major polymers that are used, synthetic polymers that are used in the food industry, food packaging, food industry, will be starting from LDPE, HDPE, and LLDPE. Okay, so there's low density polyethylene, high density polyethylene, and very low density polyethylene. Okay, or very low VLDP, very low density polyethylene, and polystyrene, PS. So you see those ice cream cups, for example, those ice cream cups are basically the polystyrene. Okay, now these materials, they will take hundreds and hundreds of years to degrade in the soil. Okay, when you just dispose it in the soil, they will take at least 500 years, 600 years, 700 years to degrade. Okay, and that is actually coming back to us via the cycles. So if we talk about if you see these papers published in different journals like Environmental International, Nano Impact, like discovery and quantification of plastic particle pollution in human blood. So people have found microplastics and nanoplastics in human blood, for example. The ingested and nano-sized and micro-sized polystyrene particles surpasses the intestinal barrier and accumulates in the body. Okay, so there is accumulation of plastics in the human body. The maternal exposure to polystyrene nanoplastics causes brain abnormalities and progenies. Okay, so causing brain abnormalities. And microplastics in seafoods, for example. Seafoods are also a major sources of contamination. But the marine environment, the marine population is under a lot of threat because of microplastics and nanoplastics. So seafood, because seafood is a big part of the food industry, there's microplastics in seafoods, implications for food security, food safety, and human health. So with all that background, you can see that this is a global problem, global issues, and we need to really work on that. So with that, we come to our approaches. So we talk about bioactive food packaging. So bioactive food packaging, when we talk about food packaging, again, the package material is very important because you cannot sell the product, the end product of the food industry just as it is, right? We have to use a package. So how about we package, but the package has some sense. So the package can really interact with the food. Okay. So bioactive, when you talk about bioactive food packaging, it will have 4 different aspects. It will have biodegradation property. It can be used as an edible coating. It will have good barrier properties, and it will have good active properties. Okay. So we talk about biodegradation. Of course, the synthetic plastics won't be biodegradable, but they will not biodegrade. But these materials, which are made from agricultural byproducts, they will biodegrade very fast. Okay. So that is one. That's one of the more major reason why we want to focus and move towards bioactive or biodegradable packaging. That can be used as edible coatings as well. So there can be a world we can imagine that, okay, we have a food, but along with the food, people can also eat the package. There is no need of throwing the package as a waste, right? So you can just directly consume the product, the food, as well as with the package. So that is where we are trying to, food scientists are already working in the edible coatings area. One classic example is already there. If you take the example of ice cream, if you take the ice cream scoop in a cone, that cone is actually from starch, right? That's mostly from corn starch. So you eat that cone, right? So that is also it's a classic example of edible coatings, which is already there in the food industry for a long time. Then we talk about barrier properties. We talk about films and coatings. So these packaging materials, they have to have very good barrier properties against UV light, because UV light is detrimental for food quality. It will damage the food, especially with the vitamins and also other molecules. And then gas barrier and moisture barrier. So you don't want oxygen to enter your system, enter into your food or interact with the food. And you don't want moisture to interact with the food, which can really impact the quality. Okay, so the packaging material has to have good barrier properties. And it also has to have good active properties. Okay, active, so that means, So in addition to all these, we can also make these packaging materials extra smart. We can make them extra smart by just adding or incorporating different classes of molecules, which are very common in the food industry. In food process engineering or food industry, they are common. So we give two types of examples. The one is the antioxidants, and the second one is the antimicrobial compounds. Okay, so antioxidants, as we know that we nowadays we are focused on nutrition. We talk about antioxidants, right? So antioxidants are those which will reduce the impact of the free radicals in the body. Because we don't want free radical creation in the body. Whenever we are having junk food, whenever you are having foods which are highly processed, whenever we are having foods which are highly treated or deep fat fried, deep oil cooked fried foods, we are generating antioxidants in our body. Antioxidants, as you know, they are not good because they will really impact at the DNA level. Because you are all biologists, you know this much better than me, but they can really degrade the quality of the DNA, and they can interact with the DNA, causing mutations and other things. So therefore, we want to reduce the effect of antioxidants. We want to reduce the effect of free radicals in the body, and that's why we're going to focus on antioxidants. So antioxidants, and of course, you can keep the food safe by focusing on natural antimicrobial compounds from different sources. So that is active properties or bioactive properties. Okay, so that is the goal and the focus of bioactive food packaging. What we are doing in our lab. Okay, again, this is just a theory of what different classes of active packaging systems are there in the food industry. We have something called oxygen absorbers. Okay, again, oxygen is bad for the food, so we're going to protect the food from oxygen. So you can have different types of oxygen embedded pads, for example, or catalyst is catalyst or metallic salts, which can be used as oxygen absorbers, which have different applications like in breads and cakes and biscuits and pizza, pasta, cheese products, and these type of things. Okay, you can have carbon dioxide absorbers or emitters. Depending on the application, it can be used in coffee or fresh meat and fishes, sponge cakes, for example. You have ethylene absorbers, for example, which can be used in fruits and vegetables, because ethylene, controlling the concentration of, because botany students, they might be knowing that controlling the ethylene concentration is very important in the ripening of the fruits and vegetables. Then, of course, we have the antimicrobial packaging or antimicrobials, which can different classes of antimicrobials are there for the food scientists, starting from organic acids to silver to zeolite, then spices, herb extracts like mostly the essential oils. Then we have BHA, BHT as antioxidants, which can all be used as embedded within the polymer for antimicrobial packaging functions. Then you have ethanol emitters, then you have moisture absorbers, flavor odor absorbers, self-healing and self-cooling systems. You can also have packaging material which can change the gas permeability. As time passes, the gas permeability of the package can change, which can be used for fruits and vegetables. Again, so all of them are possible. They are the technologies which are available to a food packaging technologist for designing the food package, or the bioactive food packages. But the focus of our goal is focused consistently on the antimicrobial side. Okay, so we're talking about antimicrobial, and recently we have started to work a bit more on the antioxidants, but we'll talk about antimicrobials first. So antimicrobial compounds or food packaging, they will act to reduce or inhibit or retard the growth of the microbes. Okay, so there are different ways or technologies to do that, like addition of sachets or pads inside the package, or it can also incorporation of volatile and non-volatile antimicrobial agents directly into the polymers. That is what we are doing. We're trying to encapsulate volatile and non-volatile antimicrobial compounds directly into the biopolymers. But you can also have coating or absorbing these antimicrobials onto the polymer surface. You can have antimicrobial polymer surfaces. You can also do some immobilization techniques to polymers of antimicrobials to polymers by ionic or covalent linkages. And you can also use polymers that are inherently antimicrobial. So in the food industry, in our food science and food process engineering food industry, we have one particular biopolymer that is really good as a natural antimicrobial, and that is chitosan. Chitosan, maybe you've heard, is obtained from seafood, mostly from shrimps and lobsters, these type of things, prawns. And that chitosan is a very good polymer that is inherently positive. The other ones you have to make it positive, but chitosan is naturally, inherently, they are highly antimicrobial, and it's also positively charged. Okay, so with that information, we'll talk about the world of sustainable biopolymers. So as a food scientist, we have a lot of options in terms of sustainable biopolymers. But when we talk about biopolymers, we are mostly focusing on polysaccharides and proteins. Okay, lipids are not really classified as polymers, even though many people call lipids as, but lipids are not really polymers, right? So we are talking about polymers here, hardcore polymers, and the source is biological, so biopolymers. So we have starches, like different sources of starches, starting from sago starch, rice starch, corn starch, potato starch, pea starch, lotus stem starch, different categories of starches. We have different cellulose and cellulose derivatives like cellulose, then methyl cellulose, hydroxypropyl cellulose, hydroxypropyl methyl cellulose, and then we have chitosan, and then we have gums and hydrocolloids. Okay, then we have the whole world of gums and hydrocolloids, starting off from guar gum, gum arabic, gum tragacanth, gum gatti, gum karaya, and then there is xanthan gum, there is curdlan gum, and so many different types of gums that all of them have different applications in the food, as well as we can also use them, explore them for food packaging functions, for food packaging applications. Whereas in the world of proteins, we have the milk proteins, one of the major sources of proteins. We have two classes of milk proteins. Just we have caseins and whey proteins. By the way, the rasgulla that we have is basically, it's a mass of casein. It's a casein micelles which makes up our rasgulla product. Okay, and then we have the soy protein isolates, and then we have zein, and then caffeine from sorghum, zein from corn. We have rice proteins, pea protein isolates, and potato proteins, and all, and enormous amount of other proteins are there, which can all be used and explored for food packaging functions. Then we talk about this same type of polymers. We're talking about the molecules are same, but the sources are different. We're talking about from agricultural byproducts. Okay, so previously we were talking about the biopolymers itself, but because the focus of our lab is more towards the agricultural byproducts and co-products, the waste, so we got to explore those materials from the byproducts. Okay, so we have different categories. We have fruit byproducts, we have vegetable byproducts, grain byproducts, legumes and pulses byproducts, and some miscellaneous byproducts. There can be many more, but we have some idea over here, okay, which will give you some ideas. So mango, for example, the mango kernel, the mango peel, the mango leaf have different categories of biopolymers, especially is the starch. Okay, then we talk about pomegranate peel extracts, pomace, grape, citrus, apple, berries, jambalo fruits. Okay, the different categories of fruits which is found in South America, for example. And there are vegetables like mushrooms and tomatoes, potatoes, carrots, eggplants. We have different categories of biopolymers from these extracts. We have grains in the wheat, for example. Rice is a major source of biopolymers. Maize, major source of biopolymer. The zein, mostly the zein proteins are coming from maize or corn. Then you have the legumes and pulses, different sources like peanuts and chickpeas and black soybeans. And we have miscellaneous categories of byproducts, starting from coffee to tamarind, because the focus of our lab is tamarind, so we are talking a lot about tamarind. Then we have cotton, lavenders, pines, and gromwells, and meadow plants, and so many different categories. Okay, so all of them have very good sources of biopolymers which can be extracted and explored in bioactive food packaging, development of bioactive food packaging systems. So with that, so because the focus of our discussion today is starch, we talk about starch and we talk about xyloglucans from tamarind seeds, and starch from other sources like jackfruit seeds or lychee seeds, for example. We'll talk about those two particular case studies. So we talk about starch briefly because maybe most of you, of course, you know about starch, right? So starch is a very important biopolymer for the food scientists because of course starch, we know that it's a storage carbohydrate in the plants, and it gives us all the nutrition that we need in terms of the nutrition. Of course, it gives us nutrition, but the starch is also important in terms of functional properties. So when we talk about the food system, we have to also talk about the functional property of the food. Okay, the food is stabilized because of certain of these macromolecules. Okay, so starch plays very important roles in the stabilization in the food processing. Okay, so again, with just the brief chemistry of starch, we all know that they are made up of linear chains. So there are two main fractions of starch: the amylose fraction and the amylopectin fraction. Again, the amylose fraction, the linear fraction is made up of around 500 to 2000 linear units of glucose alpha D chains linked by alpha 1,4 bonds. And if you see the geometry of the architecture of the molecule, it is actually in the form of a helical. So it's a helical structure. Okay, it forms a helix, but unlike your DNA, this is a single helix. And you have, so what happens is in this helix, the hydroxyl groups, because these are made up of alpha D glucose, right? So there are multiple hydroxyl groups. So hydroxyl groups are located outside. They are mostly located outside. Okay, the outside. That is how the geometry is. But if the hydroxyl groups which are more hydrophilic, so the hydrophilic side goes outside, the internal core becomes more hydrophobic. And therefore you can. So what I'm trying to say is this amylose rich starches, they are used, or amylose itself, they are used to interact with hydrophobic compounds of food, which are important in foods. For example, flavor compounds. Of course, we consume a food because it has a good flavor. It gives us a flavor. By the way, flavor is a combination of two things. It is a combination of aroma and taste. Okay, so flavor, any food which has a flavor, it's a combination of two types of compounds. It has a combination of tasting compounds plus aroma compounds. Okay, so when you talk about flavor, mostly we are talking about these are hydrophobic compounds, especially hydrophobic and lipophilic compounds. So therefore, the amylose chains can be used for interactions with these flavor compounds, and they can be used to form amylose flavor complexes. Okay, and they can be used for different applications in food, for example. Okay, so this is the amylose we all know. And then, of course, you have the amylopectin, which is the branch fraction of the starch. Of course, there is the alpha-1,6, which is the branching is because of the alpha-1,6 bonds, right? We all know that this alpha-1,6 bonds. There's a linear bond and this alpha-1,6. So if you have to see the structure of amylopectin, you have to, this N, if you see it over here, the N is equal 22 to 28. That means you have to travel 20, so you have to basically put 22 to 28 or 30 units of alpha-D glucose, and then the branching starts. Okay, so basically that's how it is. So if we compare that structure, this structure of amylose and amylopectin starch with our glycogen, on the other hand, glycogen is another molecule, right, which is the stored carbohydrates in animal cells. The molecular density of starch is much lower than the molecular density of glycogen because of this particular branching pattern. The branch density is amylopectin is less actually than the branching pattern in glycogen. That's why amylopectin or starch has a less density. Okay, so because now this monomeric units is quite high, it's 22 to 28 over here. But that's overall about amylopectin. So that amylose and amylopectin comes together to give the structure of starch. But on the other hand, if you talk about xyloglucans, which are the major structural, so there's a difference. The starch will give you the nutrition. Of course, starch also gives us functional properties that is there, but starch is primarily used for nutrition. But xyloglucans are not used for nutrition. Xyloglucans are primarily they are used for functional properties in the food. Okay, so you don't get nutrition from xyloglucan. So in any carbohydrate, so again in the food chemistry, what we call that any molecules which will, especially in the carbohydrates, the starch will be the starch polysaccharides or glucosidic polysaccharides, and then you have the other categories of non-starch polysaccharides, including xyloglucans, which will be classified as something known as DF or dietary fibers. Any molecules, any macromolecules, carbohydrates, which will not be digested by our small intestine enzymes, which will reach the large intestine unmodified, and at the large intestine they will be degraded and fermented by the microbes present in the colon to produce short chain fatty acids, SCFAs or short chain fatty acids. Those will be classified as dietary fibers. So now the whole world is going towards dietary fiber because we don't want to take starch. If you want to reduce, we're going to be healthy, we're going to reduce the amount of starch, we're going to increase the concentration in our food, the amount of your dietary fiber. So xyloglucan is one of those dietary fibers which can be a good source of this dietary fibers. Okay, so if you see the structure of dietary fibers, it's mostly linear. Okay, it's mostly linear, and basically it's a replica almost of cellulose. Okay, again, starch, as you see the structure of starch was hyper branched. This is hyper branched structure of starch. Also, it's called a branch on branch. It's hyper branched or branch on branch of starch. But on the other hand, xyloglucans, they are almost like cellulose because cellulose is linear. Cellulose is almost linear. Okay, it is not branched. There's no branches in cellulose. But the difference between cellulose and starch is cellulose is made up of beta-D glucose. There's a beta anomer that forms the cellulose. So xyloglucans are very similar to that of cellulose because if you see the chain, there's a central chain. It's almost linear, and then you have there is substitutions with galactose. This basically with galactose and mannose. Okay, xylose. Okay, so there are three monosaccharides which are used for this substitution of the beta-D glucose. That is, you have xylose, you have galactose, and you have this mannose. Okay, mannose. So these three, and also sometimes fucose, if you see fucose. Okay, so those four will be used for substitution, but the overall structure is very linear as that of cellulose. Okay, so with that information, we'll go to our first case study of our work of our lab. We're going to try to prepare this bioactive food packaging systems from seed starches. Okay, we're going to focus on lychee seed starch. Okay, lychee seed, there's not much of application of lychee seeds. We just throw away that seed, right? So how about we take that seed and focus on the starches extracted from lychee seeds, and focus on the xyloglucans obtained from tamarind seeds or tamarind kernels, and then we focus on something called lignin nanoparticles. Again, lignins are used as nanoparticles. We'll talk about that, and they are used to improve the shelf life of banana. We're going to improve the shelf life of banana, because banana, as we all know, is a very problematic food. It has a lot of shelf life issues with post-harvest quality is very poor. Okay. So this work, we just focus on lignins. So lignins are also classified as carbohydrates. Okay. So when we talk about food, carbohydrates is quite big. Carbohydrates is big, right? Starting off from monosaccharides to disaccharides, oligosaccharides, polysaccharides, and then you have dietary fibers and lignins. Okay. So all these, so lignin is also classified as carbohydrates, but the monomeric unit of lignin is quite different from the others. The monomeric unit of lignin, there is no alpha, there is no glucose, there is no monosaccharides. What we talk about monosaccharides, there's no monosaccharides over here, but it's mostly aromatic compounds. Okay, so lignins are polymers of aromatic compounds. But as an analytical, or as a chemist or biochemist or food chemist, they all classify lignin also as a part of within the carbohydrates. Okay, so lignin is a biopolymer that binds cellulose and hemicellulose fibers and provides stiffness to plants. Okay, we all know that, right? So the plant cells, they are stiff or they are stable because of three compounds. So what are those? You have cellulose, hemicellulose, and pectins. Very important. Cellulose, hemicellulose, and pectins. Those three, they interplay different chemistry that keeps the plant cells active. Okay, and then lignins also play a very important vital role in the stability of the plant cells. So lignin is the second most abundant biopolymer after cellulose, which has approximately 3 to 10 to the power 11. I'm sorry with that. It's 10 to the power 11 metric tons of lignin, and overall, if you see the structure is almost a 3D structure, amorphous aromatic polymer of phenolic nature, whose composition varies according to the plant species. Okay, so what we do over here is we try to prepare bioactive films or coating systems by a most standard and easy, simple technique that is called a solvent casting technique. And we try to prepare this film. So first film, we try to have this LSS, that is lychee seed starch alone. Then the second film, we have lychee seed starch with xyloglucan 1:1 ratio. And the third one, film C, is LSS XG LNP5, that is 5% of lignin nanoparticles, then 10% of lignin nanoparticles, and 15% of lignin nanoparticles. Okay, now when we talk about these bioactive packaging materials, one of the very important criteria we have to see is the surface hydrophobicity. We want to have the surface as hydrophobic as possible. Okay? So therefore, for the hydrophobicity, we have a standard technique which is called a contact angle measurement. We use a goniometer or a surface analyzer, also a goniometer, which is used to measure the surface hydrophobicity. So as you see, the higher the value of contact angle, that means the more hydrophobic the surface is. We want to have more hydrophobic surfaces. Why hydrophobic? Because they want to repel the water molecule, right? So therefore, it starts from 47.3 and it increases to 80.07, which is significantly different compared to the control. So we also do some raw material characterizations, like we use FTIR to characterize our raw material, the raw xyloglucan, and the LSS, the lychee seed starch. We do our as well as for the lignin, these nanoparticles and the lignin itself, because the lignin nanoparticles was prepared from the lignins, which we bought chemically from Sigma. So we did the modification, this FTIR characterizations. We also did the FESM characterization. It's FESM characterization of the lignin nanoparticles. We did particle size distribution, and we also did TEM, the transmission electron microscopy of the lignin nanoparticles. Okay, because we wanted to understand the morphology of the lignin nanoparticles and how they're distributed. Okay. Then we have, there are standard engineering techniques called rheology, because once we have to form the film, first we have to prepare the emulsion, and from the emulsion we go to the film. So therefore we have to characterize some properties of the emulsions, which we study using rheology or rheometer. And then we do the basic characterizations, the molecular level characterizations for FTIR and XRD. So FTIR will give us the functional group and the functional properties of the films. And then XRD will give us the crystallinity and amorphous nature, how much is the crystalline and amorphous properties of the films. As we see, as we increase this LNP percentages, the XC, that is the percent crystallinity, was also calculated. It was increased from 15 to 48%, and then to 47, and 44.8%. Then these are this field emission scanning electron microscopy, the FESM images of the profiles of the films. both the surface and the cross sections of this bio-nanocomposites. Then the physical properties, like starting from thickness, is a very important parameter of the food packaging material. We talk about moisture contents. As you can see that there is a initial increase, and there is almost a very similar. There's not much of a difference with the addition of the LNPs basically. And water solubility is also very important because we want the films to be as resistant to water. We don't want the materials to degrade very fast. Okay, so water solubility range from 26% to 33%, then 30, 25, and 23%. Contact angle, we know that with the addition of nanoparticles, it really improved from 47.3 to 80 degree, which is very high. And we also saw L star, A star, B star is actually their color parameters. Okay, so when in the food industry and the food arena, in the food science domain, the color of the food becomes very important. Okay, so because as a consumer, I'm not going to accept a product which is the color is not acceptable, right? So I have to accept the product with a good optical property. So therefore, the same thing applies for the packaging material as well. So for the packaging material, the color properties or optical properties becomes very important. So there are different scales of color measurement. One of the common scale of measurement is called LAP value. So L stands for L. If L is 0, there is almost dark, that is black color. If L is 100, it is almost totally 100% white color. Okay, A stands for greenness and blueness color, and B star stands for yellowness and blueness color. Okay, so that's how it changes. So there is basically a UV spectrum. It's basically a spectrophotometer which can be used for determining the color parameters. Okay, you can see the numbers from there. And these are our mechanics. So again, when you talk about packaging material, the mechanical properties of the packaging material becomes very important. We calculate something called tensile strength. As we see, as we increase our this nanoparticle concentration, tensile strength increases. And we can see the water vapor barrier property also reduces. Okay, water vapor barrier property reduces, and that means it's a good film with the increase with the this LNPs. And also, so the target of our work was to prepare these films, which are, because we are using lignin nanoparticles, to improve the antioxidant activities. Okay, so this work is focusing completely on antioxidant. Okay, so we're going to prepare films which are antioxidant. Okay, not antimicrobial, but antioxidant. So therefore, we have performed these different parameters like TPC, total phenolic concentrate, total phenolic content, DPPH scavenging activity, and FRAP assays, which are basically multiple assays to test to quantify the amount of antioxidant activity. Okay, and then finally we apply the film to the fruit for the banana coating. Okay, we apply it to the fruit. So we have three systems. We have control system, and then we have LSSXG and LSSXG. The 15% we took for day 0, day 2, day 4, and day 6. Okay, you can see how the damage is there for the quality. So for the fruits, we quantify what weight loss, hue angle, and chroma. These are all optical parameters. We try to define those parameters. Okay, define the how the application of this coating can really improve the banana quality. Okay, that's the first work, and also we also did the biodegradation studies. We did for 30 days. From day 0, day 10, day 20, day 30. We took soil samples and we embedded inside the soil sample. We put our films, and then we can see how it can really biodegrade, and we characterize the weight loss versus time. Okay. Okay, and then next briefly we'll talk about the second work, which is a more on the antimicrobial side. We focus on jackfruit seed starch, and same we keep the xyloglucans as constant over here, and we focus on zinc oxide nanoparticles. We use zinc oxide nanoparticles because zinc oxide, of course, is a metallic oxide, metallic oxide nanoparticles, but they have very powerful antimicrobial activity, and also it is approved by the US FDA can be used safely in the food. And we try to study on the application on tomato coatings. Okay. So these are again our characterization of the raw materials, our jackfruit seed starch and the tamarind kernel xyloglucans, FEACM data, and our XRD data. And then we have the rheology of the dispersions, and then we prepare these films. Okay, starting off from the control films, and then because zinc oxide material itself is white, the color is white. So as you increase the zinc oxide nanoparticle concentration, the whiteness index of the film, the whiteness value also increases. Okay, the whiteness color of the film increases. These are our different parameters like thickness content, there's moisture content, water solubility, contact angle. As you can see, contact angle, it can increase from 58 degree to 75 degree, significant increase. And the color parameters. The microstructure FEACM, we have done the FEACM. You can see really coarse and grainy structures because of the addition of zinc oxide nanoparticles at the highest level. Then our FTIR, XRD, and DSC thermograms. And these are our mechanical properties. Starting from 8 megapascal, it increases to around 20.65 megapascal. So we can really give powerful, robust mechanical property. We can improve the mechanical property of the films actually with the addition of the zinc oxide nanoparticles. And because this is a antimicrobial, of course, we're going to target microbes. Okay, we're going to use 1 gram-positive microbe and 1 bacteria, basically gram-positive bacteria and 1 gram-negative bacteria. I'm going to target how their antimicrobial efficacy is. As we see that as we increase our zinc oxide nanoparticle concentration from 1% to 3% to 5%, you can see the zone of inhibition against the microbe increases from 11 millimeter to 12 millimeter to 12.53 and 9.31, 11.37, 11.97. So there is significant difference between the control and the 0.05%. Okay, so you can because zinc oxide by nature naturally it is quite antimicrobial, and then we apply that on tomatoes. We use it these formulations for coating of tomatoes. There we have used banana, here we use tomato, and we have studied for 15 days at 3 days intervals. Again, we have 3 samples: control, JSSXG 1:1 ratio, and JSSXG with the 3%. We selected the 3% of zinc oxide nanoparticles. And we could see that we could reduce weight loss, we could almost improve the firmness values, and also we could really keep the quality, the color properties of the fruit as it is to the control. Okay. So these are our academic collaborators across many countries, from Kuwait to Canada. We have one professor from NIT Rourkela. IIT Roorkee has a good food packaging department, packaging program. From South Dakota, then lovely Professional University, Poland, this Poznan University, and Canada and Sri Lanka. And these are our funding sources who have generously supported our research at NIT Rourkela. NIT Rourkela seed grant. We have funding from ACRB Young Scientist Scheme. Then we had Indo-Sri Lanka DST's binational project, and the Spark project, which we are currently doing with Canadian universities. These are the 4 major funding sources. These are our lab students, our lab master students. They are mostly pursuing their PhD from reputed institutions like IIT Roorkee, from Washington State University, University College Cork, University of Guelph, and one student is pursuing PhD from IIT Delhi, and some couple of students working in the food industry. 2 PhDs they have one is working as a postdoc, another working as a assistant professor at Vignan University, and the current students. With that, thank you.
Thank you so much, Dr. Sarkar, for that thought-provoking and informative presentation. It's a wonderful presentation, sir.
Well, thank you.
Your insights into the sustainable use.
I hope we could hear properly, all of you. I hope everybody could hear.
Yeah, hear properly, sir. Your insights into the sustainable use of agricultural byproducts, especially tamarind kernel polysaccharides, and have given us all much to think about in terms of reducing plastic wastes and promoting sustainability in food packaging. We will now open the floor for an interactive section now. I encourage all participants to take this opportunity to ask questions or share thoughts. You can type your questions in the chat box or raise your hand to speak. So we found okay, I am just calling you accordingly. First, Ramkrishna Banerjee. Ramkrishna Banerjee, you raise your hand, please. If you have any question, please unmute yourself. First, turn on your microphone and ask your question. Ramkrishna Banerjee, if you have any question. Okay, Mahmoud Hassan. Mahmoud Hassan, do you have any question? You raise your hand also.
A question. I want to ask that
Yes.
sir told the process of biodegradable packaging, but I want to ask that it is cheaper than the casual packaging that is used now. Is that cheaper, or the processing that process, it is a vast process, no? So it needs a payment, no? So it is that cheaper from the casual one, or it is more expensive?
Unfortunately, it is not cheaper. That's the main problem. Unfortunately, it is not cheaper. The current standalone plastics that are used already in the food industry, starting from the polystyrene or LDP, HD, they are much cheaper. Okay, but if you want to improve, if you can really improve the
So
mass
that
production, then it might be cheaper. Yeah, go ahead.
so that for
Go ahead.
now we can't use that, or it is not available in all places, no, sir?
No, it is not available. In India, it's of course not available. The closest one you can get is if you buy the apples. The apples comes coated nowadays in beeswax. So there is, we can see one particular coating on the apples. That beeswax is a food grade edible lipid that can be used, that is used by the food industry nowadays. Okay, so that is food grade edible lipids. Okay, but apart from that, we don't see much of application from application standpoint. But if you go to other countries, of course, there are some materials, some packaging materials are there from biodegradable, but not much. And for that, a lot of work
Sir,
needs
I'm
to be
having
done.
one
There's a
more
lot of
question.
work Yeah.
Sir, I am having one more question that on some point you have told about edible packaging. But if we will use that edible packaging, then that edible packaging will also be harmed by the microbes, no, sir? So if we will use the edible packaging, then we have to use packaging for that edible packaging also. So how can we use that?
No,
You
so
said an
what we
example
do
that
is we
like
try
ice
to
cream cones.
Yeah. So what we do now, ice cream cone, I give you an example. Okay, that is you can call it as an edible packaging, in a way. It's not just it is just not a packaging, but it is also used for your what do you say, for improving the convenience for the consumption. But edible packaging,
Yes, sir. Yes, sir.
it's a very valid point. It's a very valid point that if you want to improve the shelf life of the package itself, then of course we have to go for antimicrobial packaging. So the polymer itself will have encapsulated natural or artificial antimicrobial compounds, which can be used for the preservation of not just the package, but also the product or the food. Okay. So of course you have to go for antimicrobial packaging. In that case, we cannot just have just starch alone. The starch alone can also be attacked by microbes. That is true. Okay.
Okay, sir. Thank you, sir. Thank you, sir. It was a great
Okay.
answer, sir. Thank you.
Thank you.
Thank you, Mahmood, for your valuable question also. And then Ramakrishna Banerjee, you also raise your hand. Please unmute first. Ramakrishna, please unmute first. Please turn on your microphone. And for the other students,
I can see the question: how do biotech-based packaging materials contribute to reducing waste? And I think Ramakrishna Banerjee asked this question.
Oh, yes.
How do biotech-based packaging? Yes, of course, you can use biotechnology. That is a very good approach. You can have biotechnological approaches for creating these biopolymers as well. Like you can use microbial fermentation. So there are certain polymers like PLA, polylactic acid, and there are a lot of in that category. There are some materials which are actually secreted by the microbes. Okay, so you can genetically engineer the microbe, and you can secrete or generate these genetically modified or engineered biopolymers, and they can also be used for packaging. So that is also one option is there. But the problem is that it will also improve the cost of the process. Till now, so our goal is to focus directly on the waste. Okay, so if you really can focus on the waste materials, then we can really bring down the cost. But apart from that, if you want to go for biotech processes, fermentation processes, for example, it also it'll improve and increase the cost a lot, right? So from a practical standpoint, we don't know how much practical it will be. But of course, in research, many people are doing that. Of course, they are doing it.
There is another question in the chat box that what current research is being done to reduce the cost of biodegradable packaging materials for mass production.
Yeah. So reduce the cost. So of course, what we are doing in the lab, that is not possible in the industry. We are just doing it for the research, for academic purpose. We want to show that okay, this type of research is possible, or this type of materials can be explored in this particular direction. But of course, if it has to be taken up by the industry, then the film production will not be done by hand, but will be done by machines. So there are certain machines like extruders. Okay, so we have extrusion process in the polymer industry, which can be used for the production of the packaging materials at a much higher. So in 1 hour, you can maybe produce 10,000 packages. In just 1 hour, you can produce 10,000 films or 20,000 films. So like that, you can use machines like extruders, which can be used for production of such materials. And then, of course, the cost will come down, right?
So I think there is another same question, Rishti Mukherjee from Rishti Mukherjee. I think you also get your answer. And the question from Preetam Gupta, he want to know about the technologies that how this process is done. That is the extraction process actually.
Yeah. So extraction is very simple process, just by water and adjusting the pH and centrifugation. It's very simple. There is no rocket science in it. It's basically a hydrophilic process. Use water and solvents and go for a certification process, and you can separate the xylem glucans out from the tamarind seeds. Very similar to starch preparation or starch extraction. Very similar. And of course, if you want more details of it, I can send you the link to the paper, and you can have a more details link of the work that we actually did. So how much concentrations and what we actually did. Okay, but process is very simple.
Last, there are a few questions. Can we take those questions? I think Shomo, we have much time in our hand.
Yes, please. Yeah, 1 or 2 questions can be done.
Okay. So there is from another question from Nusrat Begum that what are the most promising biodegradable materials currently being researched for food packaging?
Yeah, that's a good question. So if you want to develop the packaging material, you're going to focus on 2 particular aspects. You're going to focus. One is the mechanical aspect, and is the barrier aspect. Okay, so if you consider both the aspects, actually proteins have a much better future in that direction because it gives very good mechanical aspect, but it may not give the good barrier aspect. Okay, again, on the other hand, carbohydrates or polysaccharides give a good barrier aspect, but may not have a good mechanical aspect. Okay, so therefore, nowadays people are what they are trying to do is going to make composites. So protein polysaccharide composites combination. Okay, so therefore you can say both of them have good aspects, good prospects in the future, for example, as biodegradable packaging materials. But if you want to really focus on the mechanical side, you go for proteins. If you want to focus on the barrier side, you go for polysaccharides.
So I think you all got your answer. And Mahmood Hassan, again, you raise your hand. If you have any other question, you can ask.
Yes, ma'am. Apart from the topic, I want to ask that on the lowest level, what can we do to not use that biodegradable products of packaging?
I don't get it. Can you please explain what you mean?
Sir, I wanted to say that on the lowest level, means on the customer level, what can we do to not use that non-biodegradable packaging products or like that?
Okay, so at the customer level, because we don't have any option, we don't have a choice. Okay, we have to depend on plastic. We are really dependent on plastics, microplastics. Okay, so I don't see any choice as such. One way you can is, and our country is a little bit better. I would say it would have been better, but now we see the report of India Today or all these news releases. You can see that India is the largest polluter in the world. But because in our country we eat the food directly more, not like the Western countries, because the Western countries they are really dependent upon the retail chain, the retail market. But we in our country we still are not so much dependent on the retail. We still go to the local grocery and we get the grocery directly. Okay, so but if you are staying in the Western countries, for example, they are really dependent on every day. They have to depend on plastics. Okay, so one way, so that is one thing. And second is one way we can reduce is by replacing our plastic packaging material, plastic whatever grocery stores are using. For example, we can replace with paper bags, for example. So paper, because with cellulose, right? So paper bags can be a good alternative. Okay, I think government has tried a lot of times. They've tried to ban plastics, but I don't think that has worked. And therefore, now we can see that India is the leading producer in the world. So maybe paper bags, use of paper can be a good alternative.
Okay, Dr. Sarkar, we are taking the last question from our faculty, Professor Shuman Kormakar, Dr. Shuman Kormakar from Department of Botany.
Okay.
He's asking that are there any patents available related to bioplastics from the seed of tamarinds? And he has also another question that how many grams of seeds are required to make 100 grams of bioplastic?
Yeah, there are some patents available. I think there are some patents available. I have not looked into it, but of course there will be some patents available. If you want, I can look into it and send it to you by email. And the second question is what?
Second question is about how many grams of seeds are required to make 100 grams of bioplastic.
Okay, that is a good question. So I will get back to you on that. Okay, at this point I don't have the answer to it. Okay, I don't have the answer. I don't remember actually how much it is. But of course, it's a good question. I will get back to you on that. Okay.
Okay. And there is another person who raised his hand,
Yeah.
Topon Moitro. Sir,
Okay.
can you ask, please? Topon Moitro. So can you please turn on your microphone? Okay, I think there is any problem occur maybe. So I think this is the end of interactive session.
Okay.
I think some people are also they raise their hands. So Dr. Sarkar, if you write your mail address in the chat box, so they can, I think,
Okay,
ask.
sure.
Actually, we are running behind, so we have to conclude this session as early as possible and to start the next session.
Sure.
So,
Okay, thank you so much. Thank you.
Thank you, everyone. Thank you, everyone. Thank you, Dr. Sarkar, once again for this for your participation and for this wonderful journey we spent through this hour. So thank you, everyone, for participating in this interactive session. It was wonderful to hear such thoughtful questions and engaging discussions. Now, I would like to invite my colleague, the joint convener of today's event, Dr. Shoma Mukherjee, Assistant Professor, Department of Botany, to conduct the next session and introduce our second speaker. So before that, the next session, we will take a break of 1 minute only, and we will join the next session shortly.